και καλώς ήρθατε στην τρίτη διάλεξη με θέμα αρχαίο DNA που είχαμε την τιμή να έχουμε στο Ίδρυμα. Σήμερα καλεσμένη μας είναι η Εύα DASKALAKI, η οποία έχει σπουδάσει και κάνει το διδακτορικό της στο Πανεπιστήμιο της Ούψαλα σε θέματα αρχαιοβιολογίας. Και σήμερα εργάζεται σε ένα μεγάλο σκανδιναβικό project, μια διεθνή συνεργασία, το λεγόμενο Atlas Project, πολύ φιλόδοξο, το οποίο αναζητεί την καταγωγή των νεολιθικών πληθυσμών που επίκησαν την σκανδιναβική Χερσόνησο και γενικότερα αυτό που μπορώ να σας αναφέρω είναι ότι ο συγκεκριμένος τομέας στις σκανδιναβικές χώρες έχει τα τελευταία χρόνια εξαιρετικά μεγάλη ανάπτυξη, πολλά εργαστήρια με εντυπωσιακά ιδρύματα, βγάζουν τα αποτελέσματά τους σε εξαιρετικές δουλειές. Λοιπόν, δεν θα πω κάτι άλλο. Δίνω το λόγο στην Εύα. Η Εύα θα μιλήσει στα αγγλικά. Αν θελήσετε να σας απευθύνετε κάποια ερώτηση στα ελληνικά, ξέρει καλά, αλλά φαντάζομαι ότι τη δουλειά της μπορεί να την αποδώσει καλύτερα στη γλώσσα που δουλεύει.
Είναι μεγάλη μου τιμή να μιλάω είτε σήμερα και ευχαριστώ που είναι εντάξει να μιλήσω στα αγγλικά, γιατί όσο να είναι από συνήθειο, δεν είμαστε συνηθισμένοι να ακούμε τους όρους στα αγγλικά όταν δεν δουλεύει σε αυτή τη γλώσσα. Έτσι, να έχουμε μια ιδέα, να αλλάξω και γλώσσα. It's always good to give people a rough idea of what I'm going to talk about, so it's easier to follow. So my thought for today, I didn't know how much people know about ancient DNA, so I'll have a rough introduction of what is ancient DNA. And then I will, through my own work, through my thesis work, go through a historical perspective of ancient DNA. Because I had the luck of serving this field when it was fairly new. And many of the things that we're doing now was a dream when I started as a PhD student. And many of the methods are already obsolete, even though it's just been 4 years. And it sort of really shows where we started, where we are, and what is happening now. So let's start with the term, archaeological genetics. That's what we usually say when we want to be formal. Within ourselves, we usually just say ancient DNA. It's really an umbrella term. It's all sorts of genetic testing, anything we can manage to do when we get our hands on ancient material. And when I explain the difference between ancient DNA and modern genetics, I like using the analogy of a library, of seeing genetics as the genes, as books, and this is what modern genetics has to do with. Really nice books with nice text, you just open it, you read it. This is what we usually work with, if we're really lucky. This is the nice scenario. Usually, we have something like that, and add in a little bit of bacteria, a lot of dirt, and try to sift through it and find out to read something from that. The big problems of archaeological genetics are these three. That ancient old DNA is always fragmented, damaged, and heavily contaminated. And when I say fragmented, I don't just mean that it's a few pieces. The estimations are usually that, depending on the age of the sample, you have the mean length of a DNA string is 50 base pairs. In really old samples like Neolithic and Paleolithic, it can be as short as the majority being 30 base pairs. If you're among the lucky people working with permafrost samples though, like mammoths, you can have up to 150 base pairs. But those are the lucky people who work with this. This is permafrost samples. And this, but even more torn up, is samples from our area of the world. Because when it comes to these two, what we have in Greece, where we have a warm climate and a lot of exposed archaeological sites, is the worst case scenario. Our favorite place is really cold, caves that have stable temperatures, usually relatively low temperatures, and few fluctuations in humidity. And also, these can be dealt with. Technology has gone so far, we can repair DNA with enzymes. The sequencing methods we have now, they go pretty low. Even if you have modern DNA, you chop it up to sequence it. So these have become a smaller and smaller problem now. The main issue is still this. Ancient DNA is very contaminated because you lose a lot when it's degraded, and then when samples get handled, it's enough to just cough on a sample, we usually joke, and you get a lot of contamination. Even more, if you have old archaeological samples and fragmented bones, where the archaeologists used to do sort of this to check if this is a bone or a rock. They spit on it, rub it, then see if it dries fast. And you're like, okay, thank you. I really want. This is just an illustration of the main problems we deal with when we have DNA. That is a lot of add-ons and changes. So I will go on and talk a little bit more of what I have done during my years as a PhD. These two, the first two studies I've done, they are both completely obsolete. The methods I've used here will probably never be used again. And these two is the start of the future. But still, even though this work is it's still sort of already bypassed. And the reason is that, this study was based on the first pyrosequencing methods, the really old ones, before it became 454 next generation sequencing. And during this era, all we could hope for when we were working with ancient DNA was to target a single SNP. We worked with designing systems that checked 1, 2, or maximum 3 SNPs. Or as here, we targeted a part of a gene, 300 base pairs, and PCR amplifying chunks of 50 base pairs. So to target a gene of 300 base pairs, you had to run 7 PCRs, roughly. Sorry. This is just a pretty picture of the study I did to check the family relations of a Viking grave. This is some of the things we can do when we work with ancient DNA. We can check kin relationships. And this is the method I used. You see, it's just a small piece of the D loop, and we had to use 7 PCRs to amplify it. But those were the old times. Now we have moved forward, and we have the advent of next generation sequencing. Next generation sequencing revolutionized this field because we went from just looking at one gene or one SNP to actually having a hope of doing real population genetics on what we're working with. And that was a big step for us. In this study, I had samples from a Swedish warship. They are very well preserved samples. They're 300 years old, and they have been preserved in the sea. So it's like having had them in a fridge. And the scientific question, or more the archaeological question that we had, was that the archaeological data said that some of these crew members, this is Sweden in the 1700s, came from Asia. And then it's like, huh, really? People from China in a Swedish warship in the 1700s? I wonder if we can check this. Can we see if this really works? So we did the specialized DNA extractions that we do with ancient DNA. We built libraries and sequenced it, and then started the heavy bioinformatic analysis. And when you work with ancient material, this is the heaviest part. This is lab intensive, but here is where you have to check how much of this is authentic DNA. And usually we have, if it's a very good sample, we have 1% of authentic DNA. And if you have a bad sample, you can have 0.5% authentic DNA in what you're sequencing. So you get a lot of data and you can use almost nothing. So it's really amazing that we managed to get anything at all from this type of data. Nevertheless, we do manage, and we run it and we compare them with modern populations. And when you look at this plot, the axes are individual crew members. And we have extracted snips from the whole genome sequencing from these individuals. And we compare them with modern populations to see where do they fall. And for the scope of this study, it was enough to compare it with the big chunks, Europe, South Asia, East Asia, and America, to see where do they fall. Are they European or are they American? And especially the ones that are supposed to be foreign, like this individual was supposed to be South American. No, it fell squarely in Europe. And this study was mainly a proof of concept study. This is a pilot study that we intend to go deeper in. Because if we could see at this scale from where did it come, if we compare these individuals with Swedish SNP databases, we could scale in and see exactly where in Sweden do these come from. Do they come from northern Sweden? Do they come from southern Sweden? In the future, we really do aim to do this. The only thing that hinders this far is the availability of modern data. Not all modern data is available in public databases. So, but we are getting access to this and we'll probably be able to zoom in really deeply. The next study I'm going to talk a little bit more about is a Neolithic individual from Iberia. This is the first ever Spanish Iberian individual from the Neolithic age to be sequenced. And we wanted to see how does this Neolithic individual look like if we compare to others. And here I would like to talk a little bit about the Neolithic and why we find the Neolithic age so exciting in ancient DNA. Because this is probably one of the questions that most of the groups in ancient DNA are dealing with. The Neolithic transition is the turnover from the hunter-gatherer lifestyles to farming lifestyles. And this question has been an issue in archaeology for a long, long time, because this transition is the basis of our current civilization. Everything we are today, our city structures, our civilization, is based on this turnover. And we know a lot about this process. We know it, for us Europeans, started from the Near East. And then it swept over Europe in a very quick sweep for these things. Yet we do not know, was it a population movement? Was it a cultural exchange? Or was it a combination of both methods? And when we look at the archaeology, we get a nice picture and theories. When we have used modern data to look at this, we also get a really good idea. But if we really want an answer, how did this happen? And what happened during that period? We really need to zoom in and get data from the ages when it happened to see how did it happen. How did ancient population look in Greece that was near to the Middle East? How did they look in Central Europe? And how did they look in Scandinavia, which is the end of the process? So in those two ends, Scandinavia and Greece, are really two focal points of the transition. And a secondary focal point is also Iberia, because it is the end of a sweep from the Near East, but it is also an alternating route when it came from Africa. And the sample I worked with, it came from here. This is the archaeological site of Atapuerca. It's a huge cave and a very important archaeological site in this type of research, because it has been continuously inhabited from Paleolithic ages until late Neolithic and Chalcolithic age. We used similar methods as in the previous paper I used. We used next-generation sequencing, which involves building libraries, sequencing on the Illumina platform, and then heavy bioinformatic analysis. Something I neglected to mention in the previous slides when I talked about the previous study is that a big part of this analysis is to check, is this authentic DNA? And one of the ways we check it is by looking at the misincorporation patterns and the damage patterns at the ends of the sequence reads. Because when you have damaged DNA by time, and you have a fragment of approximately this length, the majority of the authentic ancient DNA sequences will be more damaged towards the ends than they are in the middle, because the ends are more exposed. If you see more damage here, or here in these two graphs, then you know that this is modern DNA contamination, because the damage is more uniformly we do the same thing as we did in the previous one. We do PCA plots, and that is, we take all the SNPs we can manage in sequence from the individuals we have, and we compare them to the big SNP databases of modern populations. And since the question in this study is a little bit different than the question I had before I was only thinking, are these European or not? Here we want to see how does this old Spaniard look if we compare them to Finnish people or Russian people that represents the northern parts of Europe here, or the Basque, that is sort of a population genetics population that we often include, or the French. And we can see that they fall squarely here with similarities to North Italians, Etruscans, and a little bit of Sardinians. A plot like this, though, that just shows one individual and how it relates to, even there are a few modern populations, doesn't tell us a lot. It tells us sort of like, yeah, it looks sort of like this. When we combine it, though, with other ancient individuals that exist in the literature, we have Swedish Scandinavian samples from different ages. We have a few other Iberian samples, but from other ages, not Neolithic. They are Mesolithic. We have the famous Iceman, Ötzi, that was available then. And then we add them to these PCA plots and see how do they cluster our ancient individuals that are the stars compared to the modern populations. And we see that all the old samples, all the hunter-gatherers, happen to be somewhere here. They cluster together. They seem to be genetically distinct from the farmers. So something happened there. The farmers were one type of population that seems to be more closely related to each other than these newer samples, because these, the blue ones and the gray, they are practically farmers. They have the new lifestyle, and this is the old lifestyle that seems to have been replaced. Still, this is just very few individuals. But a year ago, when this study was completed, this was all that was available. These were all the old ancient individuals that were whole genome sequenced a year ago. So when we did this, we were just saying, this looks really nice. This has the potential to tell us a lot. Everything that happened after that is all the fun thing in this field, because now the field is exploding. This was a year ago, just those individuals, and just in a year we are up to having about 75 to 80 whole genome sequenced ancient individuals to be published. So suddenly, from here, where we're still talking anecdotal things, yes, it was the best we could do then, but it was still, it seems like it could be, we need more data. Now we're starting to actually have the data and have the potential because we have refined the laboratory techniques. These were usually sequenced by older types of library building methods that were not as efficient as the ones we are using now. So suddenly, from sequencing one sample being a huge achievement and something we celebrated, we are looking at an era where we have papers that are presenting 60 individuals or 20, or 10 is still pretty good for this field, where you have a success rate of about 10% of the samples you have available actually giving you final sequences. And that was about everything I planned to do, a little of historical perspective, and then allow you to ask questions. Yes?
So about this relationship,
Yes.
so what's the conclusion? Was it really guy from South America?
All of them were Swedes. All of them were European, and all of them were Swedes.
Okay.
And
So
we could not zoom in to the aim when we started the study was to zoom into specific Swedish areas. But we did not get access to the database of the SNP data for Sweden. They were not enough modern samples and modern SNP data that we could get access to allow us to zoom in.
One more question. What the typical uncertainty of your data? So if you put some person to this region, I mean,
You mean at the PCA plots, because they're practically just illustrations.
so how precise this point is located? I mean, how far left, right, up, down?
It depends on the data you have available. If you have, because this plot will change a lot depending on how many, I can illustrate it better from the previous study. I have a picture. If you see here, this is a very good way of illustrating it. This individual was better preserved, so I have about, if I don't remember wrong, I have about 10 times as many SNPs for this individual as I have for this one. And as you see, it changes the accuracy of the clustering. So the more SNP you have from a sample, the more accurate your assignment is.
But still, when do you mean, when you say 10 times more, that means how many? I mean, in the range of
If you put a circle around this point.
For these ones, because these were very well preserved, we're talking at the rate of thousands. This is probably around 40,000 SNPs, and this is probably around 10,000 SNPs.
So if you put the uncertainty circle around this point, how big it will be?
Uncertainty doesn't really, it isn't really applicable in the PCA plus to have an uncertainty circle. It is, the quality of the clustering gives you an idea of the uncertainty. Like this cluster is this tight in this one that has a lot of more SNPs to work with, and it's like this. Because when you do these comparisons, you take the data you have for the ancient individual, let's say it's the 40,000 SNPs, and you only use the same SNPs to cluster the modern individuals as well. So you don't use all the available SNP data from the modern databases either. You adjust it to what you have available. Did that sort of answer your question?
Very cool. So it might be the different areas of the genome, so different loci, for example, different parts of the chromosome, support different clustering, right?
Yes.
So it might be, for example, so this PCA plot does not take this into account at all, right?
No, usually they don't. Quite often we do experiment and isolate and see if it changes the picture a lot, but it's not always informative. You usually don't include it when you actually use the data.
I mean, you could perhaps follow a bootstrap approach, taking subsamples of the SNPs from the whole genome and then find out the uncertainty area around each point.
Yes, you can do that. And we also do that for checking for authenticity data. We usually do scales of different stringencies in assessing the data and how much we include. And if you see that the data, as you said, when you do this, if the PCA plots varies a lot, then you have unreliable data. But if you do this approach and change the stringency of how many slips you include, and you see the same picture, like the same pattern, because these clusters will change spacing depending on how many slips you have. But if the clustering looks the same, despite redoing it with different subsets, then you consider it a relatively good group.
Another question concerning the Spanish
Yes.
specimen. Atapuerca is known for really paleoanthropological findings. So this kind of Neolithic is rather young, I mean very recent, and I suppose there are many other sites that one could find Neolithic and first farmer kind of samples.
There are, yes and no. There are many sites where you find Neolithic. Like if we look at the published data nowadays, we have a lot from Scandinavia that is getting a lot more thanks to the Atlas project. Then there is a lot from these areas, the central areas, because of preservation issues. And we're starting to get a lot of more data from Iberia and hopefully Greece soon as well. But these sites where you have caves that have been continuously inhabited from the Paleolithic until quite late are not as common as one might think. And quite often the samples are very, very precious. And you will not get any archaeologist that would be willing to part with Paleolithic samples easily for this type of studies, especially since the methods have been developing. So you could not guarantee to an archaeologist that give me a piece of this really precious tiny sample you have and I will give you data, because we were still optimizing. We are barely beginning to scratch the surface of actually being able to do proper genetics and proper population genetics in these samples.
So Eva, can you sum up the current hypothesis of farming moving into Europe? Is it population replacement?
Yes, it's, as always, the picture is a bit muddled. It is partially a combination, but mainly a population replacement that came from the Middle East up, combined with a little bit of leapfrog from here, where it moved in sort of jumps in Iberia in these areas where you could get from different places. And in some areas like Scandinavia, you had a parallel system. You had the parallel lifestyles, and you had the parallel populations. And you could have them living next to each other. And from what we can see from the sample we have now, they were not mixing as much genetically as one might think by populations that live in neighboring villages. So it seems to be that it was a population that came with all their culture and their ways of building things and slowly replaced the other populations and partially integrated them as well. It's never a full replacement, but more replacement with integration. It is kind of we have a lot of data from here, so we can say, well, it looks like this, it looks like we had one population, and then they look different. The big issue is that this far, we don't have much from here, from the beginning, from our areas, and we neither have a lot from here, where the farming began, so we don't actually have much to compare to. When we're comparing these, like I did in my plots, and I'm comparing to modern populations from here, we need to keep in mind that these areas have seen huge demographic fluctuations since the Neolithic. So the modern populations do not really reflect the old ones. We really need ancient samples from these areas and also from other parts of the East to compare to be able to give a better estimation. Because now we can just say, well, the populations change. They look different from hunter-gatherers to farmers. They look distinct. But we still can't say why. Did these people from here come and replace them? Or was it just chance that made the populations distinct? It's probably not chance, but it could be. Yes, please?
I don't know if it applies, but are any of the imputation techniques applicable to the domain of ancient DNA studies?
Specify imputation techniques. I'm not sure I'm following.
You need the reference genotyping
Yeah.
of a population,
Yeah.
and you have a limited set of base scores for your sample,
Yeah.
and you use that reference. What I'm really asking,
Yeah.
are there referent ancient DNAs that could help you to impute values for more SNPs for your limited SNP samples? I don't know.
There was just a paper published 1.5 months ago trying to do that, where you use imputation to increase the number of SNPs. The main issue with imputation of that type is that we're not really sure how the state of the ancient DNA would change it. And you're still making the huge assumption that this still applies, that you still have the type of structure that you have in the modern population in the ancient. It's just been done once, and it looks promising, but it needs to be done on more than a few samples they did it in, and it needs to be done so we can validate if it's useful. But it is something that is being looked into, and that could increase the strength and the power of the analysis we have right now. Any other questions?
Okay, if not, I would like to thank you, Eva, for the really exciting presentation.